All right, well, welcome again, everyone. And today we have the last Solanaceae seminar online. We are very excited about hearing about the Capsicum today. But first, I have some announcements. And first, we want to welcome Rebecca Hilgenhoff. She's here, and she's going to be the new social media manager next year, starting in 2021. She will be in charge of Twitter, Instagram, and Facebook accounts that now will be entirely focused on Sol seminars. And also, Corey Hamilton will join us next semester as a guest co-host, introducing some of the upcoming speakers. And about that, we have a new list of all the new speakers for the first semester of 2021. And that's available in our webpage. You can check there. And we are starting the next cycle of seminars on January 22nd. So just keep the date in mind, and we're going to continue with this, all these seminars. And finally, we're also very happy to announce that now our seminars will be linked with two different types of publications, thanks to the invitation of Peter Poxe. One is going to be with University Press of Helsinki for monographs, taxonomy of different groups of Solanaceae. And in second place is going to be Frontiers in Plant Science with Frontiers in Genetics. That will be a research topic focused on Solanaceae, and all the previous presenters and the upcoming presenters in these seminars are welcome to submit your results for consideration to this journal. So, and now just continue with the presenters of today, where today we have Carolina Carrizo García and Gloria Barboza. So just a short introduction, Carolina Carrizo García, she studied biology at the University of Córdoba. She also did a PhD under the guidance of Gloria Barboza. And since then, she has been working at the Multidisciplinary Institute of Plant Biology in Córdoba, where after her postdoc, she joined as a CONICET researcher. After several years working on diverse topics related to morphology and reproduction of Solanaceae, she recently focused on phylogenetic studies, especially in Capsicum. And currently, she continues working on this genus, mainly on the clade comprising the domesticated Capsicum pubescens and its closest allies, analyzing species diversity and evolution. And in second place, we also have, but she's going to start the seminars today, Gloria Barboza. And I'm very happy about presenting her. And yes, she received her biology degree at the University of Tucumán, and then her PhD at the University of Córdoba under the supervision of Armando Hunziker. Her PhD thesis was focused on morphology and taxonomy of the genus Barboza. After her graduation, she continued working with Armando Hunziker in different South American genera of Solanaceae. And just to mention some of them, she has worked in Fabiana, Genea, Diprea, Quincula, Lisiantes, and Salpicroa, focusing not only in the taxonomy, but also in the anatomy, palynology, and phytochemistry of all these genera. She has also been involved in many floristic studies for Argentina, and currently she's the co-editor of Flora de Córdoba. In the last years, she has been working on Solanum, especially the Morelloid group, in close collaboration with Sandy Knapp and Tina Sarkinen. As she's very enthusiastic in field explorations, she has long traveled through South America hunting for Solanaceae. As a professor, she has taught botany for the pharmaceutical students at National University of Córdoba for over 30 years. As well, she has been involved in many projects on medicinal plants. And since 2000, she has been focused on Capsicum, and now she's finishing the monograph of this spicy genus. So today, both Carolina and Gloria will present a complete updated framework of chili peppers through a journey exploring its diversity, taxonomy, conservation, and phylogeny. And with that, welcome both. And Gloria, you can start sharing your screen.
Okay. Can you see it?
Yes, that's perfect.
Yes. Okay. Now, thank you, Rocio, for your introduction, and also thank you to the audience for listening us in the last Solanaceae seminar of this year. Our presentation will be about 3 aspects of the genus Capsicum: taxonomy, conservation, and phylogeny. As Carolina has been working mainly on the phylogeny of Capsicum, this subject will be in charge of her. To start with, just some words on the importance of the chili peppers to complement the information given by Joseph last week. As you know, chili peppers are used worldwide as a vegetable spice, colorant in medicine and for self-defense, and are consumed daily by near a quarter of the world's population. But the importance of these peppers are in the fruits, since in some varieties they have exceptionally high levels of vitamin and so make a significant contribution to the nutritional requirements. And also because they have unique compounds known as capsicinoids, responsible for the pungency of the fruits. Coming back to the title, as you can see, we are using the word axi. So the question is, how to name these plants? Beginning with the common names, the first written common names found for these peppers is due to Diego Álvarez Chanca, who was the physician to the fleet of the second Columbus voyage. He wrote a letter in 1494 to Spain with his observations made on the West Indians. He wrote, "They," referring to the native Arawak Indian of the Hispaniola island, so they used to season it a vegetable called ají, which they also employ to give a sharp taste to the fish and such furs as they can catch and so on. So the term ají in Spanish comes from the Arawakan language axi. Today, ají, better with J, is the term most used for the pungent varieties in the Caribbean and much of South America. Pimiento is other common name for peppers, but it is also applied to other species, producing sometimes confusion to people. It comes from pimienta, the fruits of the piper nigrum, the black pepper. But it's the name for the Jamaican pepper, for the Jamaican pepper for the seeds of the allspice, pimenta dioica, and myrtaceae. Now, when the Spaniards arrived in Mexico, they heard that the ají of the Arawakans was called there in Mexico chili. Francisco Hernandez was the first European to collect plants in America, who lived in Mexico 7 years. In 1615, he wrote a book. As he was a physician, his interests were the medicinal qualities of the plants. So he not only described 7 peppers, but that were used by the Indians, but also provided the names of these peppers in the Nahuatl dialect and Aztecan language. In this way, he wrote for the first time the term chili. This terminology, chili, chile, chile, a variation of the stem chil, which means chili plants and also means red. Chile is the authentic Hispanic spelling, whereas English linguists changed the E ending by an I. So we have the English names chili or chili. And pepper is the name used in most English-speaking countries. So we can find in the literature the names alone, chili, chile, or in combination with pepper. Up to this point, these names are applied to the domesticated species. From the Nahuatl dialect, the name chiltepin has also been derived. This name, chiltepin, was given to the earliest known wild variety of peppers, which is the wild relative of Capsicum annuum. The original word in the Nahuatl dialect is Chiltepplintli. It is a concatenation of the word chile, remember that means chili plant, and tecplintli, which means flea. So the combination resulting in the flea chili, probably in allusion to the sharp biting taste of this pepper. With the time, the name changed to Chiltepin, Chilepiquin. These last two names are the most used for Capsicum annuum variedad glabrusculum in Mexico and the United States. Finally, although ají is in general in use in the territory of the Incas, the original word in Quechua, the language of the Incas, for the pepper plants is uchu or ucho, and huayca in Aymara, the other language of the Incas. Both names are still employed by some Indian groups or by Peruvian and Bolivian people, as, for example, is the case of mucuruuchu, the common name for the pepper of the Mantaro Valley in central Peru. Well, now the scientific names. I divided the taxonomical history in periods, starting with the pre-Linnean periods. 50 years after the first Columbus voyaged to the West Indies, Fuchs introduced some of the first scientifically accurate illustration of plants. He published the three first illustrations of Capsicum in 1543. But Fuchs confused these Capsicum plants with the Siliquastrum, a name that Pliny was using for the Piper nigrum, and also confused these plants with Piperitus, other name, but Pliny was using for the Guinea pepper, Aphromomum melegeta esingiberaceae. That was because Fuchs believed that these new peppers were native to India. This belief, that we know is incorrect, was perpetrated by Dodens, Bawin, Clusius, and others. It was just Joseph Turner Ford who gave in 1719 the name Capsicum for the genus and the original description for these plants, here, the name and the description, and also mentioned two origins for of the word capsicum, from the Greek captain, which means to bite in allusion to the pungency of the fruit, and from the Latin capsa, or capsula, which means referring to the shape of the fruits as the box containing the seeds inside. Well, Turner Ford listed 26 species. Well, with Linné, we have the formal names for these plants. Linné chose to maintain Turner Ford's designation of capsicum and describe formally in his binomial system the two first species for capsicum, the annual herbaceous Capsicum annum and the shrubby perennial Capsicum frutescens. Soon later, he described other two species. Then Jaquin provided a name for Capsicum chinense, Risi Pavon for Capsicum pubescens, and Villeneuve for Capsicum pendulum. So, at the end of the 18th century, the 5 domesticated species had already been described. The number of species began to increase little by little. There are many works on Capsicum describing species, but I will mention the most important, as is the first monograph for the genus, published by Fingerhut in Latin and beautifully illustrated. But again, he provided 25 species and many varieties, some of them described by himself. But again, these names are referred to the domesticated species. Concerning the wild species, one of the most remarkable works to mention is Flora Brasiliensis, where Otto Zernert recorded 10 species and many varieties. And then, of course, Dunal, in his monumental monograph for the family, accepted 50 species and again, many varieties. So, at the end of the 19th century, there were near 90 formal names given to these plants, many of them based on cultivated plant material, and what's more, based on the morphology of the fruits, resulted in a serious difficulty to understand the validity of these names. In the recent period, a new generation of taxonomists, led by Paul Smith and Charles Heiser in the United States and Armando Hunziker in Argentina, made great advances in the understanding of the genetic limits of Capsicum. I'm going to stop here to dedicate a few words to Hunziker. He was the director of the Botanical Museum of Cordoba for 50 years, where he spent his life studying primarily the Solanaceae. He had a special passion for Capsicum. As you can see here, in the mid-19th, he had described some species and had made or had proposed some combinations within the genus. For that time, the circumscription of many Solanaceae genera was somewhat chaotic, and as many Capsicum species have been described under other genera, as Witteringgia, Brachistus, Solanum, Lysiantes, Frigidardia, Hunziker decided to understand first some of them, but he didn't resolve Capsicum at all. Then he got involved in another project that was to propose a classification system for the family. That took him many, many years. And again, Capsicum was relegated. A few months before his death, his book, The Genera of Solanaceae, came out. In this book, we found the most precise circumscription for the genus. There, Hunziker proposed Capsicum as a natural group of near 20 species and some varieties, and 4 centers of diversity for the genus. But he also thought that the domesticated Capsicum frutescens and Capsicum chinense weren't independent entities, and they should be submerged under Capsicum annuum. On the other hand, he had a wide concept of some Brazilian and Andean species. So, at the end of the 20th century, there were 266 names for Capsicum in the literature and 20 accepted species by Hunziker. Before his death, he asked me to finish the monograph of Capsicum, but I knew nothing about Capsicum for that time. It took me a couple of years or more to know the problems within the genus and to understand the circumscription of the species he had accepted in his book, just studying the specimens at Court Herbarium. As I realized that the main problems were in the Brazilian species and in the Andean species, I decided to visit the herbaria and make field explorations. When visiting the herbarium, I got in touch with a local taxonomist working on Solanaceae who helped me to design local field explorations, and with some of them I had fruitful discussion on some species. Well, these explorations allowed us to understand properly the genus as a whole. And now we are in what I call the explorations through South American period that began, of course, after Hunziker's death. Well, along this time, up to last month, 15 new species have been described for the genus. In the left side, you have the species from Brazil, and in the right side are the species from the Andes. Well, after revising the 260 names assigned to Capsicum, and based on our experience in the field, we can say that the genus comprises 42 species up to the moment. Now, let's continue in our journey across the species. Just some words to remind the circumscription for the genus made by Link in her talk. As you know, the majority of the Solanaceae genera has a calyx with lobes. But in Capsicum, the lobes are completely absent, and a variable number of teeth can emerge below the entire margin. This peculiar morphology of the calyx is only shared with other genus, Lysiantes. In the stamens, the anthers opens by a longitudinal slit. Each filament broadens basally and fuses to the corolla base, forming an staminal plaque. So we have at the base of the corolla 5 staminal plaques. Between two adjacent staminal plaques, a narrow duct is formed. At the base of the ovary, there is a nectary disc that is quite difficult to observe in dry specimen, but in fresh material is very evident. As you can see here, a different coloration of the nectary in relation to the rest of the ovary, here and here. Anatomically, this nectary is also evident, this a darker zone, and produces a nectar that passes through these narrow ducts to be exposed as small droplets on the corolla limb, as you can see here, these transparent droplets. Well, the fruit. The fruit in the wild species usually is a typical berry, usually a small berry, is round, and have a persistent calyx, accrescent or slightly accrescent, and sometimes the teeth are very evident. But you know very well the high variation we can find in the fruits of the domesticated species, considering the color, the size, and the shapes of these fruits. Well, through our experience in field, we discovered the great species diversity and could select the key characters for the identification of the species. I will just mention some of these characters, the ones I use more in the key for the identification of the species. Well, the calyx, if you are trying to identify a species, you have to observe the calyx, the presence or the absence of the teeth. When the teeth are present, you have to take in consideration the shape they have as lumps or lateral compressed as a wing-like structure, or they are cylindrical or subulate, and you have to see the position they have in the flower and in the fruit. All these you are observing the flower and the fruit of the same species in all the cases. So the position in the flower and in the fruits. And when the teeth are numerous from 5 to 10, it's necessary to observe the shape, the size, the teeth have. They can be equal, subequal, or unequal, as in these cases. And we saw in one species a very interesting character in Capsicum coccinum, that not only the teeth are reflex, but also the calyx itself. Well, few species have solitary flowers. Most of the species have up to 10 flowers per node. Again, few species have more than 20 flowers per node, and we found in our last described species, Capsicum regale, an interesting inflorescence, a branched inflorescence, where each branch have many flowers that fall down easily, leaving these conspicuous scars. The position of the pedicels at anthesis is also important to observe. We can differentiate a group of species with geniculate pedicels. That means that the pedicels are upright and forms a right angle, a right or an acute angle with the longitudinal axis of the flower. And the flowers are a 90 degree in relation to the pedicels, or slightly to downward. But in the non-geniculate pedicels, of course, the geniculation is absent, the pedicels are pendent and are in line with the longitudinal axis of the flower, and the flowers hang downward. Fruiting pedicels, we notice that are also important to distinguish the species. This is the normal situation in Capsicum. The pedicels are striped, but and without a constriction between the pedicels and the calyx. And we thought that the presence of this constriction that you are seeing here was exclusive of Capsicum chinense, that allow to differentiate these species to the most closest relative. But now we know that this constriction is also present in other wild species, as happens, for example, here in Capsicum regale, and in Capsicum lanceolatum, Capsicum lisianthoides, and perhaps in other species. Well, the corollas are really beautiful. As you can see here, the color is monochromatic in some cases, white, cream, or pink, or purple, or pure yellow, and sometimes appears different kind of pigmentations. Greenish yellow pigmentations inside the corolla. In other species, this color is in combination with purple or maroon pigmentation. It happens in the white corollas or in the yellow corollas, as you can see here. And also there is a group of species where the corollas are mostly purple. The shape, most of the species have stellate corollas, but the campanulate are also present. Short campanulate corollas, long campanulate, or campanulate urceolate, as happens here in Capsicum cardenasi and also in Capsicum fribourense. And again, few species have rotate corollas. As I said before, the fruit is a small berry and round, usually round, but the color is interesting. You, in the Brazilian species, the fruits are greenish golden yellow, as you can see here, and the pericarp is very thin, is watery, and is somewhat gelatinous. In contrast, the species that grows in the Andes and in adjacent areas have orange fruits turning to red, to bright red, or to black red, and the pericarp is thicker and fleshier. And of course, here is our vedette species, Capsicum regale, with these amazing purple pedicels and purple fruits. Anatomically, we found also a character that is important, which is the presence of the so-called giant cells. These cells are already observable in the flowers. This is a longitudinal section of the ovary that, where you can see this clear zone. That is the zone where the giant cells are located. In the fruits, of course, the giant cells are more evident in the innermost layer of the pericarp. But these giant cells are not present in all the species of Capsicum. There is a group of species, particularly the species from the Andes, that where the giant cells are completely absent. As you can see here, the pericarp is homogeneous in cell size. Well, if you are trying to identify the seeds, considering the gross morphology, you have to see the color of the seed. They can be pale yellow, brown, or black. The size they have, most of the species are near 3 millimeters, but others are smaller or larger, and the shape. But if you have the possibility to observe the seed coat sculpture or structure by scanning microscope, you will really find very interesting characters that allow to identify the different species. Well, Capsicum is an American genus. This map registers all the records of the species we studied and displays the distribution along the Americas, from southern United States to Central America and the Caribbean, reaching through the Andes, Central Argentina, Paraguay, and the coast of Brazil. Laterally, you have some picture of the habitat of this species, but then Carolina will expand about the geographic regions where they grow and in relation to the clades to which these species belong. In relation to the conservation assessment, we consider the extent of occurrence, the area of occupancy, but above all, our experience in the field. So we can say that less than the half of the species are not in a threatened category, and 54 species of the species are included within a threatened category, mainly due to the endemic nature with restricted distribution and the continuing degradation of their habitat. So you can see that we have 10 vulnerable species, 7 in the endangered category and 4 in the critically endangered category. I will refer more to the critically endangered species. Well, 2 are in Brazil and the other 2 in the Andes. They are Capsicum friburgescens, is a very nice species with these purple ursulate campanulate corollas that inhabits in the Atlantic forest in Rio de Janeiro in a restricted area in Nova Friburgo. Fortunately for this species, it grows in protected areas where it forms small populations. But these protected areas faced important natural disasters in the last years, as were forest fires, great floods, and mudslides. In the case of Capsicum muticum, it's confined to central Rio de Janeiro in Serra dos Orgãos. The few collections are mostly historical and have imprecise locations for Serra dos Orgãos. The species was rediscovered in 1986 in Alto da Serra, near Petropolis, and collected several times at the same location. We went to the site and observed Capsicum muticum that is severely threatened because of the progressive decline of the habitat and the advance of the civilization to the mountain with serious deforestation of the area. As you can see here, these photos were taken last week in Alto da Serra near Petropolis. This is Capsicum muticum. Well, the other 2 species from the Andes are Capsicum benoisti and Capsicum puranum. Both were described last year. Again, both are known only by 3 locations. I visited 3 times at least the region where the holotype of Capsicum benoisti comes from, in Baños, Ecuador, but I couldn't find this species. We know nothing about the color of the flower, the fruits, and the seeds. In the case of Capsicum puranum, this species inhabits in the Piura department in unprotected areas. But the situation of this species is different than Capsicum benoisti in the sense that it can still be found in the cloudy mountain forest along the roadsides. I found some individuals along the roadsides 3 years ago. The population is small, but the forest is not quite degraded in this area. Finally, in this map, you can see the whole potential distribution of the wild species of the genus in yellow, all this area in yellow, and the protected area in green across the Americas. This scenario displays the taxonomic richness hotspots that are in darker coloration here, there. Well, these areas are outside of the current protected areas. These are especially evident along the Atlantic coast of Brazil, where the occurrence of multiple taxa overlaps. In this paper, where appeared this map and Lynn and me collaborated, We also analyzed the ex situ conservation status for all the wild species. And as a conclusion, we can say that 25 species have no reported ex situ representation in germplants databases. 8 species have fewer than 10 accessions, and 95% of the wild relative of chili peppers have high priority for further collecting for ex situ conservation. So, further conservation action is currently and urgently needed. And now, let's continue our journey with Carolina and the phylogeny.
Okay, so let me share the screen. Okay. Come on, move. Good. So let's continue the journey in this world of axi, ají, and chiles, as I like to call them. In the case of a genus like, sorry, I will move this, like Capsicum, that has some domesticated species that are economically important. To know the wild relatives of these species are valuable genetic resources. So to know the affinities of the domesticated species and to be able to identify their closest allies is really important. And this is beyond the intrinsic value of any species can have. So in this scenario of domesticated species and relevant crop wild relatives, the genetic studies and phylogenetic studies are important because they offer key information to understand the affinities of the species. And in Capsicum, the first phylogenetic analysis I will mention that use different group of species and sequences from different DNA markers. And as a result, they offer different visions of groupings of the species. You probably remember, or maybe remember, that Lynn mentioned in her talk that they recognize 3 groups within Capsicum. And the main conclusions from this analysis is that the domesticated species are resolved in the most derived clades. And there is a group of species from the Andes that Gloria already mentioned that grow in western part and northeastern parts of South America and also in Central America that are resolved in a basal clade. So after this analysis, we, in recent years, we did, okay, it had changed, hopefully. We did this phylogenetic analysis where we include, it was the broader analysis by the time. We used, include 34 species from the 35 species that were recognized at the moment, actually were recognized by Gloria as valid Capsicum species. We used sequences of these 3 DNA markers and did parsimony analysis and Bayesian inference analysis. And the results were well resolved, the trees, okay. Well, you will see all together here. And again, and the results were congruent with the previous hypothesis that the domesticated species were resolved in the most derived clades, and the Andean species here in the first splitting branch. This is maximum parsimony, and this is Bayesian inference. But there were some weaknesses. You will see really tiny here, the low supports, and some differences in the affinities of some clades. But nevertheless, we proposed this scheme of 11 clades to organize the species. Some of them were monotypic. Actually, some weaknesses led to these monotypic clades. And one case that was personally interesting is the case of Capsicum pubescens. This domesticated species is the least known among the domesticated. And there is no wild form recognized for this species, and it can interbreed with these other 2 species from the Andes, Cardinacea and Eximium, and actually these 2 species have been proposed to be the wild ancestors of pubescens. But in these reconstructions and also in analysis that we did after this one, and also in the previous analysis I show you, pubescens is always resolved separated from this species as a independent lineage. So to study better this case and also to, well, revisit all the weaknesses of this phylogeny, we did a new phylogenetic analysis, but we used a different source of data. So we used rat sequencing that worked really well. And we included in this analysis 33 species that were representative of all those 11 clades I mentioned, 55 samples. And we did maximum livelihood analysis and also Bayesian that were fully congruent and strongly resolved. And this here is a simplified scheme of that phylogeny. I have included here only one branch per species, only one sample. And I have excluded from this clade 2 domesticated because I wanted to use these 3 for other analysis and I didn't need them. And of course there are missing species because we didn't include the 42 recognized now. In general, the most clades that we recognized before were recovered except for a couple. So we have 9 clades now. And the backbone structure is a bit different. Before we found also these Andean clades splitting first, but then we have kind of a succession of clades one after the other. And now we obtain these 2 main groups after the Andean clade. Again, as in the previous analysis, the domesticated species are resolved in the latest diverging clades. Some of the weaknesses that we observed before were better resolved now, like the case of Longidentatum, is a species from the Caatinga that now is resolved together with the other species from the Caatinga, Capsicum flexosum within this big clade of the eastern species. Tobari is strongly resolved as sister to these 2 clades where the main domesticated species are included. And the triggering case, pubescens, is resolved as sister to the clade where its putative ancestors are. So this wouldn't be the Capsicum pubescens ancestor. But you will probably hear more about this next year in Nahuel's Palombo talk, I hope. Uy. Fuera. And these 2 species are, well, tricky cases because Coccineon, these 2 species before were resolved together with these 2 Bolivian species, the yellow Bolivian. But now, depending on the setting of the filtering of the data, Coccineon can be resolved here or here. And these positions also, depending on the setting of the filtering, are stronger or weaker according to the number of data. So it's something that we need to analyze better. As I said, in this structure of the genus, we have 3 main groups that we can relate to the geographic distribution of this species. In this map, all the dots are colored according to the colors I use here for the clades, except these that are also yellow, somewhere here. And well, Annum, that here is kind of pink, and the map is white. This is Galapagoense, and all these white dots are Capsicum annum variety Glabriusculum. I have excluded the domesticated species here because, well, it's difficult to say which is the native range of the species. And so we have the Andean clade that Gloria already showed in Western, Northwestern South America and Central America, and the 2 other lineages, the eastern branch, the Atlantic forest species here, Caatinga species here, with a few collections in these northern parts of South America, but these are really old, as Gloria told me. And then we have Flexuosum here in light blue. In this other branch, we found clades in Western and Northwestern South America. And some of them, these are the purple species, the Bolivian yellows, Tobari, and the species of the Bacatum clade and the Annum clade also spread to the eastern part, and the Annum, Capsicum annum variety Glabriusculum in Central America, southern parts of North America. So we see, we proposed before a sort of clockwise path of diversification across this subcontinent that we see that actually is repeated and is kind of closest here in the east again. And in this map, well, you see, and Gloria mentioned that the genus is widespread in America. It's an American genus. And looking closely at the ecoregions where these species can grow, and there are around 120 different ecoregions where the Capsicum species can be found. And those ecoregions belong to 10 biomes. And the biomes are quite diverse because we have tropical and subtropical conditions, forest, moist forest, dry forest, grasslands and savannas, and then we have temperate conditions, montane conditions, and even these extreme and opposite biomes, xeric desert and xeric shrublands, and flooded savannas, mangroves. So this is mainly in Central America, in the Caribbean. And if you see here, I have used colors to identify the biomes, and here the number of ecoregions for each species. Some species, as Puranum, that Gloria mentioned, is found in only reduced area, just one ecoregion and biome. Other species, as Rhomboideum, that are more widespread, it grows in a big number of ecoregions, but just a few biomes. Other contrasting case is Flexosum, that grows in just a few ecoregions, but not so a few biomes. And the extreme case is found in the other extreme of the tree, in these two clades where the domesticated species are resolved and actually involve the wild forms of the two domesticated species. Capsicum baccatum, variety baccatum, and Capsicum annum, variety glabriusculum. This is the most extreme case because Capsicum annum variety glabriusculum grows in almost 100 different ecoregions and in 9 of the 10 possible biomes where Capsicum species are found. And these, maybe you remember also, Leandro Giacomin talk about how much we don't know about particularly Brazil's diversity, and we see here a big hole in the sampling that maybe it's just because we have never been there. So this can change eventually. And the last thing I would like to show you is some ideas about a couple of characters, evolution, some characters that are interesting in Capsicum. And one is the base chromosome number that can be X12 or X13 in Capsicum. And they are almost evenly distributed in the number of species where they can be found, but not across the clades. This is, okay, this is the distribution of the characters of the two states, X12 and 13 in the tree. The X13 state is found only in species from two clades, the Andean clade and the Atlantic forest clade, that here is completely underrepresented. And according to this distribution of these two states, the best fitting model is one irreversible that allows changes only from 12 to 13. So we find two separate origins for the state X13 in these two clades that can be understood because of the complete different distribution of the species. The Atlantic forest is restricted to this part, and while the Andean more widespread here. And also, I'm not a specialist in this subject, but as far as I know, the morphology of the chromosome is different in these two clades, and these are smaller. Of course, these are bigger. And the genome size is also different. In the Andean species have the smallest genome in the genus, is a bit less than 2 picograms, while in this species, the values that have been registered are between 4 and 5. Last but not least, okay, if we want to move, yeah, is we find the fruit pungency, this unique feature of capsicum. Maybe you have heard about this scale, the Scoville scale, that is qualitative way to measure the quantity of capsaicinols and the presence of capsaicinols from the sweet fruits, let's say, and mild, to those extremely hot. And the pungency of the fruit is due to capsaicinoids, the production of capsaicinoids, something that Gloria already mentioned. But the product that we see, that is fruit pungent fruits or non-pungent fruits, is the result of this long biosynthetic pathway that I have simplified here, that have many steps, intermediate compounds, and several genes involved. These are some candidate genes that are the best characterized at the moment. But if we reconstruct this feature as a simple binary trait, this would be the distribution in capsicum. Let me move it, okay. Today, up to date, none species from the Andean clade has proven to have pungent fruits, and the same is for Lysianthes, while indeed across these clades, pungent fruits are dominant, even though some species like this that have all these arrows have populations that do not have pungent fruits, but that is an unstable feature. So we decided to codify it as according to the dominant characteristic. According to this distribution of the trait of the states, the best fitting model is one that allows free changes in both senses, from pungent to non-pungent fruits and backwards. So along this branch, the full machinery of the capsaicinous production will have been completed. So it's all working. And I chose to show you here the length of the branches so you can see how divergent is the Andean clade in relation to the rest of the genus. So that's it. With this picture of the fruit pungency, this unique feature attribute of capsicum that we believe is actually the feature that made them this species known to the people that eventually called them Axi. Some of these species, we finish this journey that we propose you, and thank you so much. Gloria, your turn.
Okay, thank you, Carolina. Well, now, got a moment, the acknowledgments. Well, we have to thank first our mentor, Armando Hunziker, for his legacy and for his inspiration. Of course, the team Chili Pepper, Lynn, Sandy, Ellen, for sharing with me the possibility to participate in the National Science Foundation grant to finish the monograph of Capsicum. Here we have my friends and colleagues that collaborated in the description of the species. And along this time. Also, I have to mention Eduardo Moscone, who worked in Cordoba in our institute and dedicated his life to the cytogenetic of the Solanaceae, but mainly the cytogenetic of Capsicum. And now it is continued by Maricel. Maricel participated in many works with us. And finally, we have our students who worked or are still working in different aspects of Capsicum, Victoria, Nahuel, and Vildana. And of course, in the right, we have a list, perhaps not a complete list, but of my friends and colleagues for helping us in different ways along these times. And of course, all the institutions for financial support. For all of you, thank you very much.
I'm sorry for the mess.
Thank you, Carolina and Gloria. I think my video is, yes, it's on. So yes, it was an amazing talk. Thank you about everything. So we have some comments and questions. I think the first one is Dick. If you want to turn on your microphone and video, you can ask directly. I think it was a comment at the beginning and then another one.
Well, my comment was that Charlie Heiser wrote a great essay about capsicums in his book on Solanaceae, where every chapter is a different group of Solanaceae. And if you aren't familiar with that book, look it up. It was published in 1969, but I read a portion of his capsicum essay to my class every year when I talk about Solanaceae. It's a lot of fun.
Good.
Thank you. Yeah, that's really.
Oh, can I ask a question? I want to ask about the way in which should I do it in Spanish or in English? I'll do it in English and then in Spanish because it's a question for Gloria.
Okay, okay.
Is the way in which that calyx in the recurves. So there are the teeth, and then the calyx. So at what point in the fruit development does the calyx recurve in that way? That's absolutely fascinating.
Ah, yes. You mean about Capsicum coccineum, Sandy?
Exactly. Yeah, exactly.
Yes. Well, when the fruits are still immature, the calyx is not reflex. When the fruit are completely ripe, then they turn little by little, the calyx began to recurve. It's very nice. It's a spectacular view. All the fruits together with this reflected calyx. Yes.
Well, no, because what's interesting is that happens in the old world clade of Lisianthes as well. That the business of the kind of calyx lobes being up in flower, and then you at the different stages of fruit maturity, it can be quite different. So that's super interesting.
Yes, and I saw it only in this species, only in Capsicum coccineum. So I show you this particular characteristic, in Capsicum. But for the moment, it's in just Capsicum coccineum.
Well, for sure, it happens in a couple of species of the old world clade in Lisianthes.
Very nice, very interesting.
Thank you, Sandy. Yes, that's very interesting. Gloria, you have your video off if you want to turn it on.
Oh, sorry.
Gloria, let's see you.
It's okay. We want to see you. Okay. And Sandy, you have also a comment about the Gentry work of 1992.
Yeah, 1982. So Carolina, the fact that there are no capsicums in the Amazon actually just reinforces the fact that Solanaceae is one of those Gentry families, which is most diverse in the Amazon, and in that is not diverse in the Amazon, but is diverse in the Andes in southeastern Brazil. So Gentry in 1982 wrote an absolutely seminal paper in which he described two kinds of plant families in South America. One of which had a center of species diversity in Amazonia, which is things like the Chrysobalanaceae and the Lecythidaceae, and then other families of which things like the Gesneriaceae, Begoniaceae, Solanaceae were examples, which in which the species diversity was in a ring around the Amazon, and partly that has to do with volcanic soils. But one of the things we're trying to do now is do that. Is quantify that in a more quantifiable kind of way.
Yeah, I was reading the other day about these corridors that are actually around the Amazonian, and it's more or less the shape of the diversity of Capsicum.
Well, yeah, but it's a classic Gentry family. It's a classic that paper is classic. The 1982 paper. People cite that all the time, and it's a complete classic. And so it's not a surprise that there aren't things from the Amazon.
Yeah. For me, it made sense when I read the other day this article about these corridors that follow this shape. So it fits. Yeah.
But it may not be corridors. It may be more ancient and different to corridors.
Yeah. No, really ancient. I mean, many million years.
Pebas Lake was the Amazon.
Yeah. Many million years. Yeah. Yeah.
So,
Okay, I will check that.
I think there's been a lot of debate as to whether that is an ancestral constraint on by the geography, or whether it is something that is ecologically constrained in groups. And chances are there's some of both, but
as usual. Yeah, no, so that's right. And Solanaceae is a really great family with which to kind of start a study which is comparative across families that have that kind of distribution. So say Gesneriaceae and Begoniaceae and, you know, all these kinds of families which have that circum-Amazonian distribution, and to look at the factors which are driving that distribution. So I know that's something that Lucia Lohmann is working on, looking at the Amazon, but is also true from the point of view. But you need many, many families to actually untangle that pattern, not just a single one.
Verbenaceae has that same pattern with many groups. We've been working on Lantana and Lipia most recently.
Yeah. Well, thank you, Sandy and Dick. Then we have another question from Clara Inés Orozco. I don't know if you want to turn on your microphone and ask it directly, or I can read it for you. Okay, I can read it. So Clara Inés asked, in the phylogeny work, how many species of Lysianthes were included? And in second place, some place or region in particular was Capsicum originated? So which is the ancestral area of distribution of Capsicum?
Yeah, and for the phylogeny, I used 3 to 1 Lysianthes species. I was trying in the latest. Before we used 3 Lysianthes and several Physalides, but the results are the same. It doesn't matter how many Lysianthes species I use. The reconstruction of the ancestral areas, we did it before. I was trying to do it again. I think the result won't change. Would be in the Andean region, in more or less in the region of the currently Andean species, the Andean clay species, Western South America, Ecuador, Colombia, Northern Peru, that area.
Okay, thank you, Carolina and Carines. Then there is another question of Lynn Boss. Do you want to turn on your microphone and ask it directly, or?
Sure, I'll turn on my video too, but you've blocked me.
Oh, really? Oh, it was because of the internet that there was
okay. You don't want to see me, I guess. I still can't do it.
Let me see, how can I ask to start video? It was just to keep a good internet during the talk.
Okay, here I am.
Sorry.
Yeah, my question is about Capsicum pubescens. So you guys mentioned earlier, I think Gloria was talking about the wild relative of Capsicum pubescens, and this has been a big mystery. And then on your most recent phylogenies, it looks like Capsicum eximium and Cardiaceae and one other one that are coming out on the clade with pubescens. So are we kind of settling on the fact that Cardiaceae, eximium are actually the wild relatives of pubescens, or is there something out there that we don't know about yet to be decided?
I think that there is something that we don't know yet. And I think we need to explore better the surroundings of La Paz and North La Paz and a bit South La Paz. Because there is, hi Noel, he's working with that. And unfortunately, we don't have any more money to go and sample in northern Bolivia. But we think that there has to be something, a link somewhere around La Paz, in that mixed area between dry environments and yungas, and somewhere there is a link between it.
Would you expect the wild form of Capsicum pubescens, if there is an unknown thing, to have the big fruits? Because eximium, Cardiaceae, Eschbauii have tiny fruits.
Yeah.
But pubescens, no.
Yeah, no, we have some population, we have collected some populations close to La Paz in rainforest, in the Yungas, and they have small fruits. Small fruits like
Okay.
1 point something centimeters long. They are fleshy, not dry as Eximium, Cardenaceae, and Spaugi. They are fleshy, but they are fully fleshy. It's not like the typical hollow pubescence that you have the flesh and then the hole with the seeds. These are solid, fleshy fruits and smaller flowers. And well, now probably next year we'll talk about that.
Okay, great.
So
Thanks.
that we need to explore better that area and probably find more populations similar to those. We have 3 populations, I think.
So,
Yeah, I think
so in tomatoes, the fruit size is governed by a single gene. And so it seems to me it would be really nice to kind of work with Esther Vandernap, who's giving a seminar in the next series, is she not? Who isn't spelled the same as me. She has 2 A's instead of 2 P's. But actually that could be something that would be really interesting for people who are interested in the genetic basis of fruit size. Because you can get these very, very abrupt transitions between very small fruits and very large fruits, you know, without the kind of gradual in between.
Okay, yeah. It's we can check that in the clade because the size is different, the texture is different, and then it explodes, let's say, in pubescence, big fruits, different colors and different shapes.
That's people.
I love pubescence. Sorry, I'm not being objective.
Thank you. Then we have a question from Luisa Inostroza. I don't know if you are you want to turn on your microphone and ask directly. Otherwise, I can read the question. So she said, "Good morning. Thanks for the talk. Very enriching." She's a Peruvian archaeologist, and she has been working with archaeological seeds for a few years. And the question is, have you found any difference in the thread of the 5 domesticated species?
In the thread? In the morphology?
I think she means in the morphology, or yes, in the use, yeah.
Yes, well, the point is that when you observe the seeds by naked eyes, you can see a seed coat and a smooth seed coat. But when you observe it with more magnifications, the differences among the domesticated species appear. And we can differentiate Capsicum, frutescens, chinense. Perhaps they are more similar, but Capsicum baccatum and Capsicum pubescens are different. Surely we find differences, but studying the structure and sculpture of the seed coat. The gross morphology seems more or less similar.
That's super interesting. Thank you, Gloria. Yes, well, of course, there are a lot of congratulations and a lot of comments about, yes, how useful and how complete was the storytelling. And I also have a question about have you found any correlation between color of the fruits with pungency? And is there any hypothesis about which would be the precursor? Like if it was first the color and then the pungency, or the other way around?
I'm going to trace that. It's an idea I'm developing because this the new phylogeny is a bit different from the previous one. So we have these 2 big groups, the Eastern and Western, as I grossly call them. So I think there is a play there between colors and pungency, because the origin of the pungency may be before the change of color to yellow fruits. So, but I need to work on that and reconstruct it. We'll see what the data says.
Oh, that's great. Yes, because it was. I remember when we were collecting with Gloria some capsicum in Brazil, that they were kind of pungent when they were immature. So
Immature.
yes, and then when they turn more yellow, they were no pungent anymore. So it was kind of, oh, that's super
Yeah,
good.
yeah. We'll see. We'll see what happens.
Okay, I don't know if there is any other question for Carolina.
I have one. This is Lynn.
Sure.
While we're talking about pungency, so do you guys know, or does anyone know if I'm thinking about the origin of pungency, and so Carolina's last slide showed a point on the phylogeny where pungency is hypothesized to have evolved in comparison to the non-pungent capsicums and then to Lysianthes. And one of the main genes or gene regions that's been implicated is PUN. So the PUN1 AT3 gene region. Do we know anything about the structure of that gene in the broader context of Lysianthes and capsicum? Whether it is even, it exists, or if this exists and is not expressed for some reason in the non-pungent members of that clade. Actually, we were hoping to do this in our latest grant, but for various reasons, it didn't work out.
Yeah.
Do you have any insights on that, you guys?
I was trying to amplify the PUN gene in the whole capsicum, and using the available primers, I could sequence the gene and going down more or less until the yellow Bolivian species properly. And then I had to develop new primers to sequence the rest. It works. But so far I could never amplify the PUN gene in the Andean species.
We tried to do that too with the published primers. We didn't try super hard, but we had trouble amplifying it and trouble with some products that couldn't be aligned.
Yeah, sometimes it amplifies something that is nothing because it's a big mess of things. But it's something that I also would like to explore a bit more. I know there are 83, how do you call this, ortholog sequences in other genus, in other genera.
Okay.
But the product is not the same. It's just the enzyme. So or a similar enzyme. So you need the whole biosynthetic pathway to make it work. But yeah, it's something that I couldn't finish yet, but maybe we need to try harder and how do you say, design new primers and
I'd like to see someone look at it. And I don't have time, and for various reasons, and it sounds like you're involved in other things, but this would be a wonderful project to look at those genes in the pathway and see, now that we have a really good phylogenies for the whole clade, and see what the story is of pungency. So to be continued.
Yeah,
the next generation.
Noel, that is there. You should continue this journey. we are a sort of dynasty. Gloria, Unzica, Gloria, then me, and Rocio, that is kind of Gloria's daughter, and my cousin. So maybe, why not? And Noel, the next one, so all dynasty.
No, I have too many things for the physalogs. I cannot do anything in capsicum. Thank you. No.
You've got lots of paleobotany to do, Ro.
Exactly. Yes, on top of that. So it's physalogs, paleobotany, and So Sandy, you were saying something about about the alkaloids in mature fruits and of tomato.
So there's a lot of work been done in acanthophorus, Lynn. You remember that about the fact that there are lots of high alkaloid contents in immature fruits, and then they get translocated out back into the rest of the plant at fruit maturity. So what you get is, so maybe it's a disruption of that. So maybe pungency in mature fruits, because you were saying that they were pungent when they were immature. So maybe pungency in mature fruits is actually a disruption of this translocation, of this pathway about translocating things back out of mature fruits.
Yeah.
So it's not necessarily a gain, it could be a loss. So there's been a lot of work done on acanthophorus about that.
Oh yeah, like Marty Cipollini has done a ton of stuff about fruit alkaloids and how they change over time.
So I don't see why capsaicin should be any different. So perhaps it's the same kind of thing. Is that these ones that are pungent at maturity is just a loss of something in the pathway that allows it to be translocated back into the plant to be reused again.
Yeah.
So it's not very adaptive, really.
Well, I read somewhere that the immature fruits were hotter, so the mammals won't eat them until they are ripe. And then eventually they were not so hot, and
No, but I think Josh Tewksbury has really showed with a lot of his work that has to do with microbes much more than with mammals.
Yeah.
I think Josh has shown that both of them have an effect, both the mammal deterrence and the microbe defense.
Microbes, yeah. Well, another topic to explore.
I feed my birds hot pepper suet because the rats won't eat and the squirrels won't eat the suet.
Ah, that's nice.
You have to ship me some, Dick. We can't get that here, I don't think. I'll have to make it myself.
That's nice.
And it really works.
Yeah.
Wow, that's great. Yeah. Okay. Any other question? I see that Ellen is with a video. I don't know if you have a question as well. No? Okay. So in that case, we're finishing this seminar. But before we finish, if you can all tune, yes, some people had to leave, but if you can all tune your videos, we can do a picture all together. This last seminar of the year. Let me see if I can change. Yes, gallery view. So please turn on your
everybody.
Video.
Let's go.
So we can have a full picture.
Joseph, Christine.
Yes, Clara, Inés.
Inés, come on.
Anita, Leandro. Joseph.
Andres. Bang.
Andres.
Let me try some better light here.
Gabriela, yes, Joseph.
Hello, Gloria.
Some people escape as soon as you say turn on the
people in the sunshine. I'm really jealous. It's already dark. Ro, you're in the museum still.
No, I'm still in the museum. I'm still in the museum. I don't want to leave.
Oh.
Okay.
Oh, I know.
Okay, I'm doing the printing. Okay.
Only 8 o'clock in the morning here.
Some people is escaping.
Okay.
They
Yes,
don't
I
want
have
to be
the
seen.
picture.
Bro, please email us the print. Send us the print, please.
Yes, sure. I will mail you. Yeah. So thank you, everyone, and thank you very much, Gloria and Carolina, for the talk. And so, well, and next year we are starting on January 22nd with Esther van der Naap, and she is going to talk about mining tomato germplasm for crop improvement traits. So that's
So Ro and Chelsea and Andres, I just want to congratulate you because this has been a wonderful initiative and it's been really great to get together and for everybody to exchange ideas, and just thank you so much for setting this up. We really, all of us, super appreciate it. Thank you.
Yeah.
Thank you.
Congratulations.
And thank you, everyone, because without you, there is no seminar. So thank you, everyone, for attending. And yes, and for
no organizer, it wouldn't happen.
Okay. So see you next year. Have a nice holidays, and yes, and enjoy the
Happy New Year.
Happy New Year.
Happy New Year.
Take care, please.
Take care, folks. Bye to everybody. Thanks, Rose.
Bye. Take care.
Bye.
Bye-bye.
Bye.